Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Novel Digital Endpoint Solutions for Early and Mid-Phase Development. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. So just type your questions in the chat box by selecting the questions icon located on the right-hand side of your screen. If you require any assistance, please contact me at any time by sending a message using this chat panel. I'd also like to welcome our attendees viewing this presentation from our LinkedIn Live event, and I encourage you to submit questions as well via the comments tab. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available for streaming to those who register on xtalks.com. At this point, I'd like to thank Clario, who developed the content for this presentation. Clario is a leading provider of endpoint data solutions to the clinical trials industry, generating high-quality clinical evidence for life sciences companies. They offer comprehensive evidence generation solutions that combine medical, imaging, eCOA, precision motion, cardiac, and respiratory endpoints. Now I'll put on my webcam as I invite my co-moderator in, and I will welcome our presenters. So first, I'd like to welcome Dr. Todd Rudeau, who brings strong organizational leadership as Clario's Executive Vice President and Chief Medical Officer with over 2 decades of clinical cardiology and pharmaceutical research experience focusing on drug safety. He works centers on developing scientific and regulatory strategy while advancing innovative technologies that strengthen clinical trial rigor and improve the customer, patient, and site experience. Next, I'd like to welcome Dr. Tina Sulis. She has 30-plus years of deep expertise across the pharmaceutical, biotech, and clinical research sectors. As the founder and director of Alethea Life Sciences, her distinguished career includes serving as CEO of a major CRO, director of an ASX 200 company, and VP of Clinical Strategy and Development. She is one of the few Australian clinical development leaders who has successfully taken products from lab to regulatory approval. Tina has managed clinical programs across all development phases and diverse therapeutic areas. Tina holds academic appointments at the University of Melbourne, advises US-based biotechs, and serves as an Australian resident director for international entities. She is a pivotal figure in the R&D community, frequently speaking globally on clinical innovation and industry collaboration. Next, I'd like to welcome Dr. Kelly Dumais. She brings over 15 years of scientific leadership as Senior Director and Global Head of eCOA Science and Consulting at Clario. Hello there. She drives a global strategy for eCOA development and implementation and focuses on advancing eCOA methods, improving site and participant training, and shaping PRO strategies for oncology trials. And next, I'd like to welcome Dr. Anu Banzel. There he is. He serves as Vice President of Imaging in Oncology with Clario. Dr. Banzel provides leadership and oversight of all aspects of imaging in Phase 1 to Phase 4 drug and device trials, from protocol design through registrational submission and post-approval monitoring. He uses his 20-plus years of experience in clinical research and patient care to support Clario's cross-functional teams and clients in medical, regulatory, and scientific affairs. Now, with the introductions out of the way, it is now my pleasure to pass the mic over to my co-moderator, Dr. Rudeau. So when you're ready, you may begin.
Great. Well, thank you, Sonia, for the very kind introductions. Thank you to all of you, our global audience that has joined us today as well. I'm excited about this one. It's our first webinar that we've deliberately timed to occur during working business hours for the APAC region. Equally importantly, it's our first webinar that we've dedicated to a specific regional development strategy. So that is the opportunity to leverage an Australia-initiated development strategy for a global program. So I think we're going to learn quite a bit today. Why don't you go ahead, Sonia, to the agenda slide, and we'll take it from there. We're going to take you through this ambitious agenda today. After a general introduction on the unique challenges of early phase development and how a proper digital endpoint strategy can address those challenges, I'll then turn over to Tina, who's going to talk us through the considerations for that Australia-initiated early development strategy I mentioned, and what the distinct advantages are or that can enable. From there, we'll hear from Dr. Demay with considerations around building out an early phase patient-reported outcome strategy, and then Dr. Banzel speaking to how novel approaches in medical imaging can provide actionable insights in early development, more so than traditional site-based imaging would classically allow. Tina's then going to step back in as we near the close of the session with important operational considerations, highlight some recent trends, and also include a real-world case study or two illustrating the concepts that we've covered. After a brief wrap-up, we'll do our best to leave some time for Q&A. I would highly encourage you to use the chat function. Even for those questions we don't get to at the end, we'll commit to directed follow-up via email, certainly. I could go to the next slides, and actually one more. So let's talk about early phase development and what is unique here. I think what's most relevant is that these early studies provide the data by which incredibly impactful pipeline decisions get made. The data can mean the difference between investing in one program versus another when you can't fund both, is that decision is most directly informed by the insights achieved in these phase 1B or 2A proof of concept studies. But, and this is the key challenge here, the sample sizes are small, the study durations are relatively short. And in that setting, you need very precise endpoint measurements to reliably detect a small signal within any noise. That signal of efficacy can be challenging to uncover. As mentioned, there's also short duration studies, so the endpoint selected needs to be very sensitive. For example, in an early phase, we wouldn't have an 18 or 24-month study to measure the impact of an Alzheimer's drug on cognitive decline or the time needed to assess for a meaningful change to functional status in a motor disorder study like Parkinson's. So anything we can do with technology and best practice applying that technology can really move the needle beyond just getting the typical cursory look at safety or PK, but actually learning something meaningful and informative about the drug's effects. I can go to the next slide. So given that we're after precision and accuracy in study endpoints, that leads us to my next point here. It's not just choosing the right endpoints to measure, although that's typically the focus of most development teams, but also how to measure them effectively. So I guess there's three considerations really. First is, what are we measuring, right? Tumor dimension, ECG changes, spirometry metrics perhaps. Second consideration, what technology are we going to deploy to take that measurement? And the technology, of course, impacts precision. And then third, what can we do with site and patient training or education so that the implementation of that technology we selected adheres to best practice, right? Bolstering the precision and reproducibility of measurements. For imaging, that could be about training sites for best practice in image acquisition. For cardiac, it could be about training sites on how to minimize motion artifact or autonomic effects on an ECG. For ECOA and ClinRose, it could be about placebo response training, rater training, independent review during study conduct. Again, the impact of these three considerations in early phase is especially high. So what to measure, what technology to use to measure it, and then how to optimally train sites and patients to minimize variability in how that technology is applied. Next slide. A few additional points here in the next couple of slides before turning over to Tina, starting with some considerations beyond just what you will learn if you include informative endpoints, but also how will that technology potentially impact recruitment and retention. So for example, a patient-friendly wearable as a heart rhythm monitor, one that allows for unrestricted activity, exercise, showering, it can transmit data without returning to the clinic. That's very different than another arrhythmia monitoring solution, such as a traditional wired Holter recorder. And while flexible and patient-friendly solutions are essential for recruitment and retention, there's still the issue of maintaining data quality, and that needs to be balanced for any solution that's being considered. So for example, if we're combining blood pressure data that's measured at home with blood pressure data that's measured in the clinic in the same data set, then leveraging the same model blood pressure cuff with the same inflation algorithm, electronic data capture in both settings, it's a great way to reduce noise and improve, precision. If you allow any brand or model of blood pressure cuff to be used at home, have the patient or participant physically bring in a diary to the clinic. That data is then manually, entered into the EDC system. Well, this achieves the opposite effect, right? You've achieved the ability to collect data at home, but the data quality has deteriorated, and making decisions on that data will ultimately convey more risk. The bottom of the slide here is just to very quickly highlight an early phase opportunity that we won't spend a lot of time on today, but is essential not to miss. And that's the first in human study in particular, whether that's healthy volunteer or an oncology patient. The broadest range of dosing is always administered, or almost always administered, compared with later studies in the development program. And this presents an optimal setting for assessing and modeling the effects of a drug on the QT interval at different doses. So the PK levels and the change from baseline in QT interval, they can be evaluated to assess for that relationship. And it's accomplished by recording high quality ECG data at the same time that the PK sampling will occur, and then analyzing this in a statistical model at the end of the study. I can go to the next slide. This all changed about 10 years ago with the 2015 update to the ICH E14 guidance, which enabled proper phase 1 ECG and PK data to replace a future dedicated TheraQT study in most cases. And I mention this as the method in which the data is prospectively collected and analyzed during that study is critical in order to meet the regulatory expectation. So it needs to be set up before the study is initiated to do this properly. But in the vast majority of cases, as I mentioned, when done well, millions of dollars can be saved in future development costs, avoiding a TheraQT study altogether. Plus, and this is really important, the development team gains much earlier insight as to whether the drug does in fact have a QT safety risk or not. And next, please. So this is the last slide for me. It just illustrates visually the type of modeling that I just described. Each dot is a decile of PK exposure along the x-axis. The corresponding change in QT interval is seen on the y-axis, change from baseline, that is. And ideally, this relationship is flat, as you see on the left, meaning no matter what the exposure is, high or low, the QT interval isn't changing. That's a drug that does not adversely cause QT prolongation. But on the right, you see a positive drug. And so as the exposure increases from left to right, the larger increases in QT interval are subsequently seen. Again, I point this out just as one area where the right strategy in early phase, prospectively deployed, can really build confidence in pipeline decision making, despite having relatively few patients, despite having a short study. You'll hear later from Dr. Banzal and Dr. Dumay on other important early phase strategies to gain meaningful insights like this. But first, I'm going to turn over to Tina Solis to talk to us about considerations for an Australia-initiated development strategy and what global companies should know, when considering this. So, Tina, over to you.
Thank you so much, Todd. And welcome to everybody wherever you are in the world. And a huge thank you to Clario collaborators for inviting me to be part of this really important and informative webinar. This morning, I'm going to spend this morning for me, I'm going to spend the next 10 minutes telling you a little bit about Australia and utilising Australia as a strategy, particularly in early phase projects. So next slide, please, Sonia. Before I go into Australia, I just want to set the scene by telling you a little bit about Alithia Life Sciences. Really, we are a premier Australian full-service capability but boutique clinic contract research organisation. One of the most powerful things we do is engage with clients as early as possible to provide to them a timeline and costings and scenario of what an Australia clinical trial may look like. And just like any other CRO, we are available to support contracting with phase 1 sites and clinical project management, data management, all the good things that any other CRO can offer and does offer. But where we differentiate is that every single person that works for Alithia has at least 15 to 20 years industry experience. And I'm proud to say that in 5 years of operation, we have 0 turnover of staff. We are very well known for our first-in-human studies, neurology, devices, oncology, immunology, ophthalmology, and the list goes on. And you can see from the graphs on the right that about 70% of the projects we are involved with involve small molecules or pharmaceuticals, about 20 to 25% involve devices, and the remainder are cell and gene therapies. You can also see from the graphs that we are particularly involved in phase 1 and 2 because they're the type of studies primarily coming into Australia. And we are also involved with phase 3, both as collaborators with other CROs and looking after sites in Australia. Next, please, Sonia. So one of the questions we mostly get asked early on is, particularly if clients have not worked in Australia before, you are so far away. How does this work? Yes, we do acknowledge we are far away, but what I can say is that there is a lot of overlap, particularly at the end and the start of the days, if you're in US and Europe in particular. And what we can also say is then while you are sleeping, we are working. So you will wake up to an influx of emails and a continuum of clinical trial activities. So it really does work in that sense. Next slide, please, Sonia. So why Australia? We are very well known for good quality data. We have a very strong and stable government, a very strong IP protection laws. But one of the most powerful reasons people come to Australia is because the data is acceptable to other regulatory authorities such as FDA and EMA, and we have a very ethnically diverse participant recruitment pool, which means that about 30% of anyone living in Australia have been born overseas. And that ultimately means that the data you obtain and collect as part of your clinical trial is also demonstrative of the populations in countries such as the US and in Europe. So that is you don't have to repeat your data. It's a very powerful place to be. Next slide. So we can see from an MTP clinical report from a couple of years ago, and if you look at the top graph and the last pie chart, you can see that we had a very even spread across phase 1, phase 2, and phase 3 studies. And the bottom graph demonstrates that some of the most popular or common therapeutic areas assessed in clinical trials in Australia were oncology, neurology, gastroenterology, and similar diseases. You can see it's similar of what the typical development indications around the world as well. The next slide also demonstrates that Australia has clinical trial activity across the country. It is also fair to say the most populated areas, which is mainly the eastern seaboard, and the states of Queensland, New South Wales and Victoria have the highest clinical trial activity, but really that all of Australia does undertake clinical research, particularly important in remote areas like Northern Territory, where we have some rare diseases and we have a lot of diabetes and those types of indications as well. Next slide, please. So we often, one of the most powerful aspects of why also people make a decision to come to Australia is that since 2011, our federal government introduced a very fair and generous tax incentive. That means if you're a company with a turnover of less than 20 million Australian dollars annually, you can quite easily and quickly set up a local entity or local subsidiary. It takes about a couple of hours, costs about 4,000 Australian dollars, but in exchange, anything you do that is considered research and development in Australia, you can claim. And when you put in your tax return once per year and you fill in your R&D tax incentive application, you will get cash back of 43.5%. So that, with the exchange rate, makes it up to 60% less expensive to do the same trial in Australia versus the US. Next slide, please. And one of the other important factors is that it's very quick to have a trial approved in Australia. Whether you're undertaking a phase 1, 2 or 3, a drug or device study, you never really have to have an open IND. That's a very powerful place to be. So we find companies come to us, they have their package of information just like you would anywhere else in the world. We submit to our ethics committees and they review once per month generally. Some review once per week. They will always have questions, and if we try and answer those questions within a week or two of receiving them, they will usually, in 9 out of 10 cases, approve the project out of session. Once we have IRB or ethics approval, we submit a form online to our regulators, and they acknowledge the trial. They don't review. That acknowledgement form is called clinical trial notification, and together with the ethics approval will allow us to ship investigational product on site and start recruiting for our study. We also have a very powerful mutual acceptance system, which means for single site studies, it usually takes about 60 days or 2 months from final submission to first patient, first visit, or for multi-site studies up to 3 months, but one site takes on the lead approval and the other sites recognize that approval and just undertake contracting or governance. We also have a very powerful template by Medicines Australia, which is a clinical trial agreement template, which means there is really not any toing and froing between legal departments, also saving time. Next slide, please. So we often get asked, what information do we need to go into clinical trial in Australia? Just like anywhere around the world, we need a protocol, investigator brochure, informed consent, some clinical trial insurance, anything patient-facing, such as the ECOAs, patient-reported outcomes. We do need to label our investigational product slightly different. According to Annex 13, we need an expiry date, but we also need a local sponsor. If you are setting up a local subsidiary, you can be the local sponsor. If not, you can delegate local sponsorship to a CRO such as ourselves. Next slide, please, Sonia. And this last slide before I hand back to my colleagues is just reiterating the timelines and what's involved. So I won't cover this to a big extent, but really preclinical means any preclinical information that you'll need to go into clinical trial in the US, with the exception of long-term toxicity. And then we have up to 3 months for startup. The project duration goes from first patient, first visit to last patient, first visit. And the last 3 months of any project are usually to clean up and lock the database, provide the tables, listings, and figures, close out the sites, and write the clinical study report. So I'm going to hand over to my colleagues now.
Great. Thank you very much, Tina. And thank you all for being here. I'm excited to be here. I'm going to switch gears a bit now and talk about the value of incorporating subjective patient experiences in early phase trials and how this can better prepare you for later phase success. So what we're hearing more and more from regulatory agencies is the importance of incorporating that patient voice into drug development programs. Most notably, both the EMA and the FDA have guidances on this, including the FORA guidance series, the patient-focused drug development series the FDA has been releasing. So how do we incorporate the patient voice? Most traditionally and formally, that's through the use of patient-reported outcomes. And these are right to measure disease symptoms, treatment effects, quality of life directly from the patient. So if using PROs to support, especially if you're planning to support them in your trial as primary, secondary endpoints, there are many benefits to starting as early as possible, so including PROs in early phase trials. And there's a few reasons for this. So first, it's the first way to really ensure patient centricity, in that it's the first time that you can incorporate the PROs formally in your clinical development program. And PROs are really the gold standard method for measuring subjective experiences. So especially when disease symptoms or treatment effects are known to impact quality of life, PROs should be used. Secondly, it's important to optimize, or it's an opportunity to optimize your later phase PRO strategy. So early detection of, for example, side effects or improvement in symptoms or quality of life will allow you to better focus your PRO strategy for phase 2 and 3, potentially also revealing opportunities for drug differentiation, especially in a highly competitive market. Thirdly, it will allow you to be submission ready. So we know that with options for accelerated approvals, especially in oncology or rare disease, treatments can be approved in non-phase 3 trials. So incorporating PROs in early phase will allow you to be in line with regulatory expectations. And then finally there, including PROs allows you to also better evaluate drug tolerability, which we know is one of the major objectives of early phase and phase 1 trials. This is a point that leans a little more specifically to oncology studies, as there has been a lot of recent literature and regulatory guidance around the usefulness of PROs in early phase oncology studies. So this is kind of my segue into oncology as a case example for using PROs in early phase studies. Next slide, please. So you can see the FDA supports the use of PROs in early phase dose-finding oncology trials in this guidance. This guidance was finalized in 2024, and it lists PROs as recommended data that should be used as part of optimizing your dosage strategy. So it states that PROs can provide a systematic and quantitative assessment of expected symptomatic side effects and their impact on function, and should be considered to enhance the assessment of tolerability. So note that PROs are not replacing clinician-reported tolerability or safety data in early phase, but it is complementing that data, giving more granular, detailed data on the patient experience, and specifically how symptoms and side effects impact functioning in daily life, or their ability to function in daily life, as this is really the level of detail and granularity that is often missed in clinician reports of symptomatic adverse events alone. Next slide, please. So to dive into this concept of tolerability a little bit more, I love to show this article. It's not a new publication. It's about 15 years old now, but I think it very nicely shows the potential pitfalls of relying solely on clinician-reported data for assessment of side effects and tolerability, again, particularly using oncology as an example. So current traditional practice for collecting adverse events and toxicities in oncology assumes that clinicians have an accurate picture of the patient's subjective experiences. But we know that this isn't always the case in reality. There's really no standardized method of how clinicians are assessing AEs across sites, across studies, and across visits. So this study here directly compared clinician-reported AEs with patient-reported patient symptom reports. And what they found was that clinicians tended to systematically downgrade the severity of patients' symptomatic adverse events, detect symptoms later, and then even miss symptoms altogether, especially those really subjective experiences that, again, are really best known by the subject, like mood or nausea, etc. There is also a paper that was just released last month or the month before that looked again at inter-rater reliability of clinician AEs with or without them reviewing PRO data. And what they found was that clinicians that had access to PRO data consistently, their consistency of grading AE significantly improved. So again, just a nice confirmation of the recommendations around the importance of incorporating PROs of the patient voice in early phase dose finding trials. Next slide, please. And this is also showing that it's not just the FDA recommending early phase PROs or a couple of isolated studies supporting it, but that there is true international consensus around the benefit of incorporating PROs in early phase studies. Again, the focus of this paper was oncology trials, but here with this paper, there's a couple papers listed here. What they conclude is that there are both general consensus and agreement among stakeholders, including patient advocates, industry representatives, academic researchers, pro-methodologists, regulators as well, and that PROs should be reviewed when making these sorts of dose escalation decisions and determining the maximum tolerated dose and the recommended phase 2 dose. So while there are some operational considerations, which I'll briefly get to next, there is true, clear, agreed upon benefit of using PROs in these phase 1 trial settings, including that they can aid clinicians, right, in AE reporting, can detect early AEs to inform dose strategies, could even minimize dose modification or treatment discontinuation later on. So again, kind of true consensus is being had around the benefit of incorporating PROs in early phase trials. Next slide, please. So now when it comes to kind of operationalizing PROs in early phase studies, I'd like to outline just quickly some of the regulatory thinking around expectations for data quality standards. So the age-old, do you use paper or ECOA, electronic clinical outcome assessments for PROs, especially when thinking in early phase. So at face value, PROs administered on paper might seem like the easiest solution, but there are a lot of considerations and risks around the quality when using paper. So global regulatory bodies confirm, right, that electronic source data provides high quality data. It avoids parking lot syndrome, which is when a patient just fills in all their diaries or questionnaires in the parking lot right before the visit. Regulators also state how electronic data collection can increase compliance, reduce variability, reduce missing data, et cetera. And then there's even a couple of oncology PRO specific guidances there on the right that also specifically call out the benefits of eCOA versus paper for oncology trials, stating that it can reduce patient burden, missing data, et cetera. Next slide, please. And then this gives even more details and examples around the risks of data quality when using paper COAs. So for example, when using paper, there's increased risk for transcription errors, as that paper still needs to be manually entered into electronic database. There is higher risk for missing data, as I mentioned, inaccurate and inconsistent data, backfilling by patients. There's increased site burden with not only having to do data entry and source verification, but also needing to ensure that all of the assessments are completed in full by the patient and in the right order according to the protocol. There's also manual scoring calculations that can, again, add to site burden and also lead to errors. So on the other hand, eCOA can minimize or eliminate these problems by using things like reminders and notifications to help with compliance and participant engagement. Set windows of diary availability ensures that pros are done at the right time and in the right visit and in the right order. Edit checks, even at the point of data entry, can also reduce conflicting or inaccurate data, for example, flagging an entry that is illogical or not possible, or automatically skipping over questions that don't need to be completed based on previous responses. So bottom line is that, eCOA can really help both ensure that the data is cleaner at the point of entry with on-device edit checks, but also with the use of notifications and site notifications for compliance. It can be a way to proactively address kind of concerning data trends before they even become issues. Next slide, please. And this was a recent publication that came out last year that shows even independent evidence supporting the use of eCOA in early phase oncology. So this study included weekly remote PROs and found 85+% compliance for the pros, showing high acceptability even among patients that were older or who had more advanced cancer. It also showed that pros captured 3 times more symptoms as compared to the clinician-reported symptomatic adverse event reporting, CTCAE. Pros are also identified as or the pros identified key symptoms that were commonly missed by clinicians. So again, kind of just overall supporting the use of using pros in early phase, but also supporting the use of eCOA, ePros in early phase oncology trials as well. Next slide, please. And this is my last slide just to show how we implement eCOA. So this shows the many different options that are available when implementing electronic PROs in early phase or in any phase of a study. The key here is really that you want to make sure that the technology is flexible and easy to use so that it easily fits into the patient's lifestyles and expectations for how technology should work. The exact device strategy, so whether that's a tablet, provision device, or maybe patients use their own device, BYOD, bring your own device. There's a number of considerations that go into the device strategy, including where the pros are being completed, the patient population, geographical considerations, et cetera. So our teams of experts are also here to kind of help with identifying the right PRO strategy and also the right eCOA strategy for collecting those PROs. So that's all I have time for my section. I hope that was helpful for you in thinking about incorporating pros in early phase development programs, and I will hand it off now to Anu. Thank you.
Hi, thanks, Kelly. Good afternoon, good evening, good morning, everyone. I'm going to use my time today to talk to you about why I think it's important to centralize imaging in early phase development programs. This really extends across all therapeutic areas. We're going to start on the next slide by talking a little bit about how this applies across neuroscience, across general medicine, GI, oncology. And then as Kelly did, we're going to hone in a little bit on oncology as sort of a case study, partly because we have a limited amount of time today, and also because more than half of all of our new trial starts in early phase are in oncology. So we felt like it would have the broadest appeal. So what is the value proposition here? Well, I mean, centralizing imaging provides several really important attributes. It allows you to harmonize your site imaging protocols, which increases the quality of your data. It also avoids data loss. Whenever we do a retrospective trial, we see that we never collect more than 85% of the data. Doing this prospectively really allows you to have a curated data set that's of high value. It provides you the opportunity for additional ad hoc analyses, whether those are done prospectively or retrospectively. It provides you objective data that you can provide to larger pharma partners that you might want to work with in later stages of clinical development. There are other aspects as well. When you move away from efficacy and you talk about safety monitoring, perhaps in an Alzheimer's trial, it's essential to have really good safety monitoring early so that you can detect any safety signals that might exist. And the best way to do that is really to centralize it. If we move on now and just try to dive in a little bit more specifically into oncology on the next slide, one of the things I'm not going to try to convince you of is that the value of centralizing imaging in early oncology is because you can get a high quality RECIST readout. We all know that RECIST is flawed. In addition, you can get RECIST data from the sites, and most of the time this data is not used for regulatory purposes anyways. So what is the real value? Let's move on to the next slide. Before we get too far into this, I want to talk about just define a couple of things that we're going to spend more time on later. Just they may be self-evident, but just to make sure we're all on the same page starting out. So there's the concept of total tumor burden. What is that? Typically, that's meant as volumetric segmentation of all of the measurable tumor lesions that are visible within a participant's body on imaging. You can see here on the left, we have an image. This is AI-generated segmentation of all the lesions. Why haven't we been doing this historically? Well, you can imagine if you needed a radiologist to draw, to contour each of these lesions on every slice throughout the entire chest, for example, it would take hours. And it's just been prohibitively expensive and logistically infeasible. One of the true tangible benefits of AI, there's a lot of touted benefits that are sometimes hard to recognize, but AI-based segmentation has really made a big difference in what we can do. And we'll talk a little bit about the value of tumor burden, but in my mind, the real value of total tumor burden is that it facilitates all kinds of other advanced analyses that are incredibly powerful. One of these that we'll dive a little bit deeper into is called tumor growth kinetics. This is in essence mathematically modeling the rate of change of tumor size over time. With that, we can go to the next slide. I said I'd talk a little bit about what total tumor burden in and of itself could be used for. There's a lot of things that, conceptually some of the easier things are balancing treatment arms, for example, making sure you have an even distribution of total tumor burden across multiple treatment arms, which you can't really do if you're using RECIST and measuring 5 lesions, right? You can identify high-risk subgroups where standard tumor medication doses may be insufficient. You could even bake this into inclusion or exclusion criteria. Next slide, please. As I had sort of already highlighted, the real value to me of the total tumor burden is the fact that having those volumetric measurements of all the measurable lesions allows you to do all kinds of other things. One of them, as I pointed out, was tumor growth kinetics. So what is that? In the solid black line, you can see here, this line is representing behavior of the tumor volume over time. So as you might expect, a patient may start a new medication at day 0, and over time, that tumor volume will decrease as the medication works, and then it will slowly but exponentially start to increase in size until you hit 20% and the patient has progressive disease. Using different mathematical modeling techniques, however, you can break that tumor behavior of decline and growth into different components. There are multiple components. The two easiest to really conceptualize in my mind are tumor growth and tumor decay. If you want to look at tumor growth, it's this dashed line, starts in the lower left corner and extends exponentially up to the right side of the screen. And then there's tumor decay, which is the dot dashed line, starts in the upper left and then slowly flattens out towards baseline. When you combine those two elements, you get the overall tumor behavior. Why is that important? Let me just make it a little less abstract. If you take that G constant, the G score of growth, that can directly be converted into tumor doubling time. So you could literally say that treatment A has a tumor doubling time of 250 days, and treatment B has a tumor doubling time of 500 days. So just, again, to make it a little abstract and not talk about G's and things like that. If we go to the next slide, please. What is the point of this doubling time? What's the value here? I'm going to try to point out three different attributes here. First, the G value is very highly correlated with radiographic progression-free survival, as well as actual overall survival. This G value correlates better with overall survival than ORR does. I mean, think about that for a second, right? The metric all of us have used for more than 20 years to determine a drug's efficacy is less correlative to overall survival than a different value that's derived from the exact same data set. And then finally, and this one's really interesting, this G value can help predict response and survival both earlier in a trial and with fewer patients. So again, if we're looking for signals of early efficacy, this is perhaps one of the answers that might help answer that. Go to the next slide. I made three pretty bold statements. I'm going to try to show you some data that supports these. The next three slides are all from the same paper. What they did is they had a large phase 3 breast cancer trial with over 500 patients because it was powered for RECIST. They retrospectively calculated G values and looked at how that data broke out. So in terms of G correlating with survival, this is a plot that shows G stratified by quartile. And as you can see, the lowest quartile in purple in both treatments, there's one treatment arm on the left and a different one on the right. And you can see that survival in the lowest quartile in both treatment arms is much worse than the lower quartile. And you can actually even see that the second lowest quartile is different between the two treatment arms, where it's much more similar to the other quartiles on the right side. We go to the next slide. We can see some data that supports the fact that this growth constant correlates better with overall survival than a RECIST-derived overall response rate does. You can see the RECIST-derived response on the left with tumor response. The concordance with survival is 0.72. And when you look at the G value on the right, the concordance is 0.82. So significantly higher with the G value. Next slide. And finally, this is really interesting. Again, remember, each of these two arms, in the blue and the red, were powered with over 250 subjects in order to be able to show a difference by RECIST. When you look at the first, on the far left, you look at the first 40 patients that were on trial for at least 14 weeks, you can see that there's not that much difference between the two treatment arms, right? If we look at the horizontal black bars, they're showing a relatively similar G-score, which again converts to tumor doubling time. However, when you go to the middle graph, you look at the first 60 patients that have been on trial for 14 weeks. Now you see a pretty big difference in the G value between the two treatment arms. And what's even more interesting is that when you look at the first 80 patients at 14 weeks on trial, and that follows through, there's other data in the paper about the first 100 patients, the first 120 patients, that difference in G value is maintained, but it's not really changed. If you think about it, what it means is that you're getting a readout within the first 25% of patients enrolled in 14 weeks that correlates directly with the RECIST outcome you get at the end of the trial. Can we go to the next slide, please? There are some other things that G does. We are short of time. I want to make sure we get to Dr. Solis's case studies. So I'll just briefly mention some of these. Dose finding, we often, if we do get centralized imaging, it's usually during a dose expansion phase, but why not use it during dose escalation? Again, you need very small numbers of patients to be able to see differences in G. Imagine if you could see that your second highest dose and your highest dose have the exact same G value. This allows you to move away from max tolerated dose. It helps satisfy the FDA's Project Optimus mandate, and it, in the era of combining drug therapies, why would you want to use a higher dose than you need to? Referential versus experimental, I think I'm going to skip. It's really just about trying to utilize imaging data from pre-enrollment scans to compare to on-study scans rather than just using baseline data. And then in terms of event prediction, in addition to response, you can also predict progression events. Next slide. This one I'm going to do quickly. It's just a little bit of in-house data. This was actually, we were not actually trying to figure out how to predict. We were actually just trying to answer a different question about which mathematical modules to use. But we took some non-small cell lung cancer patients that had a central readout already, so they had 2D ROIs placed by radiologists. We converted those into 3D volumetric ROIs using a similar algorithm to the one I showed earlier. And then we used the first 4 on-study time points to predict both progression and response at week 30. And we can see is even with this small a data set, we were able to predict progression with more than 70% accuracy and response with more than 80% accuracy. To go to my last slide, really the message I want to leave you with, we did a deep dive on oncology because that's really what we had time for. But the message I want to leave you with is centralizing imaging in early phase trials is really essential across all therapeutic areas, whether it's neuroscience, gen med, GI, they all have different features like this and different types of advanced analysis. And if you're not having a partner do some of these types of analyses for you, you're leaving a lot of really valuable decision-making data on the table. With that, I'll hand it back to Dr. Solis, take us through some case studies. Thanks.
Thank you very much, Anu and Kelly, really important information. I'm going to use the next 10 minutes or so to take you back to Australia and bring back all the good information we've just learned and a little bit more about the type of trials that we are seeing coming into Australia and the trends. Next slide, please. So right now, the type of studies that we're seeing coming into Australia is your traditional healthy volunteer, single ascending, multiple ascending dose. But what we are seeing that's becoming very common now is in the same protocol, the same database, the same ethics committee approval is a MAD plus cohort, a cohort of patients with the indication that a particular trial company may be interested in. That cohort may be as small as 3 to 6 participants or patients, or it could be as big as 32 and 40 participants. Either way, it's a really powerful adaptive trial design. We're also seeing first-in-human oncology studies start in Australia without an open IND, and those particular companies undertake their via ethics approval, their dose ranging studies, and then their dose expansion in US using an open IND. We are seeing definitely complexity of studies, the patient burden and assessments. And examples would be that even in early phase 1 studies, we're seeing the use of EEGs, as well as, of course, ECGs, as we've heard, CSF sampling multiple times, imaging, of course, central imaging still important, and of course, genetic definition of participants, even in the healthy volunteer stage. We're definitely seeing an increase of wearable and analysis of biomarkers, and the wearables will differentiate, that data differentiates your product compared to others in the market, and we are seeing biomarkers via wearables for cognition, for sleep, for movement, for voice. We're seeing an increase of vendors such as working with targeted trial recruitment vendors, particularly in rare diseases, where we also work with patient advocacy groups. We are seeing home visits increase, particularly those involving infusions, and direct-to-patient delivery of study drug. We're seeing specialist CROs, not only boutique CROs like Alithia, but CROs that specialize in oncology and radiopharmaceuticals and neuroscience, for example. An increase in private sites. You may recall Australia has for the past 40 years Medicare, and that means that everyone is able to access very quickly and freely public hospitals, but we are now seeing an increase of groups of private sites that can decrease those startup timelines by 3 to 4 weeks as well. And also, we routinely now work with patient advocacy groups and clinical trial networks who are able to quickly and efficiently work out the feasibility of a project, patient populations, engagement of sites and key opinion leaders, and work at the per patient budget. Next slide, please, Sonia. I want to also talk about, we've heard, and I just want to bring it all back to the biomarkers and how important they are, both from a regulatory perspective. We've heard from both Anu and Kelly. We know FDA very serious about biomarkers. We are definitely seeing the examples that I've listed on this slide. And what we always say to our clients, particularly even in a single ascending, multiple ascending dose, if you are not sure and you're still an early stage company and you're developing your biomarkers, we also recommend that you put a general statement in the protocol that says you are going to collect biomarkers, particularly for blood and urine and other biomarkers for future development of biomarkers. So next slide. Really important. We've already heard extensive discussion by Kelly. Thank you, Kelly, about patient-reported outcomes. Bringing it all back again, that there are definitely general FDA guidances that have been around for a while now. And if you want to develop a particular patient-reported outcome, either with collaborators like Clario, with others, you do need to go back to the guidance, especially if you, and validating the patient population, especially if you then want to include that information on your target product profile and on the label of your product. So important to consider validated systems and using partners like Clario to support you in these endeavours from day one. It's not too early to consider from phase 1 onwards. Next slide, please. One of the biggest changes we've seen in clinical trials in Australia, particularly since COVID, is that of decentralised clinical trials, particularly home-based visits. We now have routinely do these visits, particularly in between visits. We collect data, we've spoken about that, using different tools, wearables. We do have e-consent. We deliver product to contactless, direct to the participant's home, therefore decreasing the burden. And that means in a country like Australia, which is geographically huge, but population small, we can access a diverse range of participants wherever they may be. Next slide, please. I do want to also acknowledge we've embraced AI in Australia. We're still working it out, but from a day-to-day tool at Alithia, we use it for note takers. We use it to undertake review of our protocols and the likely succession and refining of the protocol. But want to also acknowledge that whilst we have a federal healthcare system in Australia, the hospitals are run state to state. So there is a little bit of a discord, but what I can tell you is that we have pretty consistent access to records via electronic medical records, and when you are setting up a trial and you want to monitor that data, that is available consistently in Australia. Next slide, please. First in human sites exist all around Australia, and you can see the stars denote the locations. Many of these sites are Clario accredited sites. All of these sites are qualified vendors on Alithia front. But it means that they can all work with you. We have at least 6 dedicated phase 1 facilities, which means over 400 beds available for true first-in-human studies, in addition to the first-in-human oncology sites that are available all throughout the public hospitals and private centres in Australia as well. Next slide, please. I wanted to point out oncology is one of the strengths in Australia, and one of the site management organisations called Cancer Trials Australia, our partners as well, released their annual report late last year showing the trends have stayed consistent with regards to number of clinical trials, especially by commercial sponsors in Australia. And you can see most of the indications in oncology in Australia are in haematology and also solid tumours. A shout out to solid tumour group called Omico, where they can actually pinpoint exactly where every single patient with a solid tumour is in Australia, and we work with them to work out which sites are then feasible for your study. But you can see the other cancers are also very well represented in Australia, and the numbers of those projects have stayed steady in the past 2 years. Next slide, please. Okay, just a quick note to say that it's a very commonplace strategy for studies to start in Australia without an IND, for companies to use that information and go back to the FDA and open up an IND. Next slide, please. Just conscious of time. And I want to move to the case studies. All right, the first case study is that of a biotech working in oncology. We do work with experts that have oncology first-in-human committees to refine your protocol, particularly making it feasible in Australia. We work with the site management organisations who will prepare ethics, site budgets, take care of annual reports and your budget going forward for all the sites. We start off these type of projects without an IND within 4 months from final protocol to first patient, first visit. We work with partners like Clario. Dose escalation is undertaken in Australia. That information is then taken back to open up an IND. And the continuation of partners like Clario for imaging, for central imaging, is taken through to the whole study. And the data originating in Australia is perfectly acceptable to the FDA. And on top of that, companies like this open up a local subsidiary and are able to claim all of these R&D initiatives and costs under the R&D tax incentive on top of the exchange rate. And my last slide. Another example is in a first-in-human Alzheimer's disease study, another US biotech. Again, we use clinical trial networks like Dementia Trials Australia, about 26 sites across Australia and New Zealand, who will work with the sites to undertake feasibility, negotiate the per patient budget on behalf of all of the sites, and undertake contracting. Again, from final protocol, first patient, first visit, we took about 3 months, working with digital partners for the central imaging. The company was able to use this data to go to a pre-IND meeting in the US and open up an IND and then expand their clinical program. As well as that, they were of course able to set up a local subsidiary and claim all of their costs under the R&D tax incentive. So I might leave it there to allow a few minutes for questions. Thank you very much.
Great. Well, certainly thank you, Tina. Thanks to Drs. Benzel, Dr. Dumais as well for your insights today. Really interesting content, and appreciate those case studies at the end as well. I hope you all, our audience, found this valuable. I think there's really two sort of key themes that you heard about today. One is the understanding of the nuances in selecting an endpoint strategy and all the potential benefits that this can afford you as you make decisions at the end of that early phase program. And then now, specifically understanding the considerations around applying those strategies within an Australia-initiated development paradigm and all of the nuance that Tina was able to share with us as well. From a Q&A perspective, obviously we are out of time. We have some great questions that have come in, and I do promise to each of you we are going to follow up directed by email to each of you to have your questions answered. Sonia, maybe I'll invite the other speakers to come back on screen just as we close out here, and Sonia, I'll have you come on as well and close us out. Thanks.
Okay. Well, thank you very much for that great presentation. It was a very fascinating, and I'm sure everybody enjoyed it. Just like Dr. Rideau said, if we could not attend to all your questions, the team will follow up with you after this presentation. Now, if you just take a look here, if you're interested in hearing more from Clario, you can register for their next webinar, Modern Approach to Multiple Sclerosis Trials, Applying Innovative Digital Endpoint Solutions, scheduled for Tuesday, March the 24th, and I put the link in the chat box already there. And then I'm also letting you know that I've also shared with you today's webinar link, so please go ahead and share that with your colleagues. So thank you very much, everyone, for participating in today's webinar. As I mentioned, you will be receiving a follow-up email from X-Talks with access to the recorded archive. A survey window will be popping up on your screen. Your participation is appreciated, as it will help us to improve on our further webinars. Okay, and for those of you watching via LinkedIn Live events, we encourage you to visit xtalks.com to register for this event and gain access to features such as viewing the recording and more. Now, please join us in thanking our speakers for their time here today and for Dr. Rideau for being my co-moderator for today's event. We hope you found this webinar informative. Oh, one more thing. Forgot to share the email addresses and the contact information here. So please, if you have any further questions, please utilize the email addresses you see there on your screen. So thank you very much, everyone, for being with us today. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. Until next time, please take care and bye for now. Bye, everyone.
All right, thanks a lot, guys.
Bye. Thank you.